Ok. Hola a todos. Hello, everybody. Gracias por la invitación. Estoy muy contento de participar en este webinar. Voy a charlar en inglés, pero mi presentación es en español. So let's start. So the main topic today is related to carotenoids, and of course, natural carotenoids in that case. With a product called Panaferd, and we'll mention the two main effects of Panaferd: color, of course, but also salud, health of shrimp. So it's interesting and important to mention, of course, main sources of carotenoids in the wild, in nature. You have mostly four of them: bacteria. You have also yeast, you have algae, and sometimes bacteria, cyanobacteria in that case. Bacteria is, you got one picture of here showing actually Paracoccus carotenifaciens, which is a source of Panaferd. So only these four microorganisms are able to produce natural carotenoids, which will be then eaten by what we call primary consumers, of course, zooplankton, as you well known, but also, of course, some fish, some echinoderms, so bivalves. So then these primary consumers might be then eaten as well, but what we call secondary consumers, of course, a major of fish, some cephalopods as well, and including shrimps. So of course, this is a cycle, what we call the food chain, pretty well known in nature. Some general consideration regarding carotenoids. Of course, as I said, no animals, and especially large animals like fish or shrimps, may metabolize carotenoids. It's important to remind that there's some confusion sometimes. Most carotenoids are considered lipophilic, so they trend to store and to be stocked in more fatty tissues. They are mostly absorbed by a visible spectrum. So this is a HPLC and chromatograph image showing between 400 and 500 nanometers. You see astaxanthin, contaxanthin, and the other main carotenoids. Of course, it's a large family of colors, from yellow to really red and deep red colors. We'll see that later on. Carotenoids are divided in two major families. The first one having carbons and hydrogens, and the main family, and we'll talk more about it, which are xanthophylls, containing carbons, hydrogen, and of course oxygen, which is the main character and specific difference with the carotenoids. Let's talk briefly about the main functions of carotenoids, especially for shrimp. You have four main functions. The first one is related to colors. The second one is related to what we call photoprotection, or UV protection in that case. The second one is related to immunity and especially antioxidant activity. And the fourth one is related to, let's say, precursor of vitamin A or pro-vitamin A, which is retinol activity in that case. So these carotenoids will help in different situations for the shrimp, and especially related to adapt the shrimp to some very stressful conditions they may face in the ponds or in the wild. Of course, salinity variations, especially, of course, during rainfalls, salinity may drop heavily. You have also heavy lights, especially during the sunshine. You may face oxygen depletion, especially during dawn or heavy bloom. You have pH, of course, effect. You have sometimes very high temperature or very low temperature, which is not so good for the growth. And of course, some pathogens. So all these types of stress, carotenoids will help the shrimp to adapt to these various conditions. We'll talk more in details later on about the potential effect of carotenoids. So why we have carotenoids? And you have some images here showing where you can find carotenoids, especially in the aquatic world. Of course, salmon is well known, and you have here a very deep salmon, Atlantic salmon, using Panaferd, which is the natural color we'll talk about. You have this sea urchin, and you see the very deep ova, red ova, colored by mostly astaxanthin in that case. Of course, you may find shrimp and crabs, and especially ova as well here. So the main interest is, of course, to protect the species, and that's the main reason why carotenoids are stored by the animals. For shrimps, as you may know, some of you, carotenoids are stored in some specific cells, what we call chromatophore, and all the carotenoids, and you have here a picture of chromatophore where you can see the red deep astaxanthin color. So this is typically the types of cells you will find especially in the exoskeleton, both in the body or in the head. So it will be just below the epiderm. So it will protect the shrimp against pathogen, as we said, of course, including bacteria, fungi, or virus, but including as well, and we'll see later on, some injuries. So this is a very specific indicator of stress, I would say. And you all know melanosis problems. And here you have a picture with, you can see the black spot of melanosis, which is a very specific indicator of stress, and of course, degradation and catabolism of carotenoids. So of course, according to conditions, carotenoids may not be found in the same quantity. So of course, in the wild and in some live food, especially zooplankton, artemia, or copepods, you will find carotenoids in certain amount and certain quantity. But of course, according to the conditions, especially as we said, with low temperature, especially with low oxygen, you may face some difficulties to get a good bloom and a good plankton development, and it will affect the carotenoid content. And you see here this graph in the same type of ponds in that case, but according to the density, you see that in very extensive conditions, shrimp will find enough carotenoids in the wild, in nature. But of course, in very super intensive system, and you got more and more of this type of forms in LATAM or in Asia, of course, the quantity of carotenoids will not be enough to sustain the color and sustain the immunity of the shrimp. And of course, it must be added to the feed. So that's the interest of this natural source of carotenoids, and including this bacteria. We'll talk more about it, and naming Paracoccus carotenificens and brand name of Panaferd. So you will find in the wild various form of carotenoids. And here you have a table showing Panarus vaname, white leg shrimp, coming from the wild. And you see in different parts of the shrimp, from hepatopancreas, exoskeleton, or muscle, the different forms of carotenoids. Astaxanthin, as I mentioned, is a main form for shrimp. But you may find as well contaxanthin, beta-carotene, zeaxanthin. Mostly astaxanthin is found in exoskeleton, including hepatopancreas. It could be found in two forms. Chemically, that's what we call free. And the other point is related to esterified, which can be also another major form for shrimp. As you can see here, which is interesting to mention, in the hepatopancreas, so where the head is, you find a very high level of esterified astaxanthin, 144 micrograms per gram in that case. So especially during the molting phases, of course, when the shrimp will molt, they will lose this part of the cuticles, and the hepatopancreas will release in the next part of the new exoskeleton the astaxanthin and the carotenoids needed to get the color and to protect the animal. It's a bit complex, this slide. I won't go too much in details, but it's showing you, of course, the different pathway of carotenoids from beta-carotene at the top of this graph down to astaxanthin. And this pathway are very important, and you can see, and we got the confirmation that most of the shrimp are able to use different pathway from adonirubin, which can be found in Panaferd, then to astaxanthin, or from adonixanthin, also found in Panaferd, going up to astaxanthin. So it's very interesting to understand how the shrimp will use these different forms of carotenoids. And that's also the interest of natural forms, to get a various wide spectrum of products. So some words, of course, about Panaferd. What's Panaferd? As I said, it's a microorganism. It's very tiny form, about 0.2 micrometers. It's a bacteria. It's found mostly in the soils or between the soil and the water. And it can be said it's the start of the food chain. So it's a very important microorganism for all the aquatic chain. What's interesting with the natural carotenoids that you will find different and let's say three very important nutrients here. Of course, you will find carotenoids. We'll talk about that later on and more in details. But you have also, of course, micronutrients, especially phosphorus. You have also zinc, which is very important for shrimp. You have calcium as well, magnesium, a bit of iron. So that's the type of micronutrients that also the shrimp can use when you include Panaferd. What's also interesting, it's micronutrients, which is not found, of course, in synthetic forms, as it is a natural microorganism, a bacteria. The cell walls will contain, of course, proteins, but also lipids, but also some polysaccharides. I won't go too much in details today about polysaccharides contained in this bacteria, but it could be very interesting in terms of immunity. A few words about the production methods and process. So it's quite easy. So we got a sample from a bank we got, very detailed Panaferd banks. So we use fermentation in a quite easy way with a specific media with water. Then after a few hours of bacteria development, we remove fully the liquid and the media. and then dry the product. So as you can see, it's fully natural, nothing is added, and no, of course, it's not GMOs, there's no chemical, no antioxidant added in this type of product. A more detailed profile of carotenoids found in Panaferd. This is a very, as I said, wide spectrum from astaxanthin to adonirubin. Maybe a few of you know adonirubin. This is also very interesting and red carotenoids found, for example, in some shrimps and including crayfish. Canthaxanthin, adonixanthin, these four major, let's say, red carotenoids are the keys of Panaferd product. The last ones are more yellow forms in that case, but as I show you, the pathway are possible to move from beta-carotene up to astaxanthin. What is interesting to mention here is that in Panaferd, we'll find what we call very rare, very unusual carotenoids and not found in synthetic forms, and especially adonirubin and adonixanthin with astaxanthin, and astaxanthin is by far the most well-known carotenoids. And they have very powerful antioxidant activity that we'll see much more in details later on. It's very powerful for three various reasons I want to mention now. As I said, they are found in free forms. Again, they are not esterified, which can be the case in some microalgae. For example, Hematococcus is an esterified form. So it will be much easier for the animal to use the carotenoids, and they don't have to digest and cut the carotenoids between the ester and the carbon forms. It's a trans form and not cis, which is also considered as more powerful in terms of antioxidant activity. The optical form, it's also isomeric form, it's also what we call SS form, which is also quite different and found in nature compared to the synthetic astaxanthin, which is a mix, a blend of racemic forms, not found in nature. So a few words about antioxidant activity. So as I said, this table is interesting because it compares the different activity or antiox activity from vitamin C to vitamin E to lutein. Lutein is quite known and found, for example, in corn. Lycopene, which also found mostly in tomatoes. So as you can see, it compares on a base of 100 the different antioxidant power, and you could mention that here adonirubin and adonixanthin has the same or much higher astaxanthin antioxidant activity compared to the others. And of course, by far much more than vitamin E and vitamin C. So that's pretty interesting, and that's the reason why Panaferd is so powerful for shrimp. Another point, it's much more technical here. It shows what could be the antioxidant activity of the both adonirubin and adonixanthin, especially in terms of what we call the single oxygen activity and power. And again, on a 100 or 1 scale, adonirubin and adonixanthin have shown very powerful antioxidant and single oxygen quenching. Regarding, again, the inhibition of lipid peroxidation, which can be the case, especially when you store fish or when you store shrimp, these three red carotenoids, asta, adonirubin, and adoxanthin, show very interesting inhibition of oxidation of lipids. This slide is just confirming the difference between macronutrients and micronutrients, phosphorus content, calcium, iron, magnesium, and very interesting high level of protein, which is quite unusual, above 50%. So it could be also, in terms of nutrition and formulation, taken in account when you formulate a feed. Few words about the difference between Panaferd and the synthetic astaxanthin. It can be compared sometimes. That's why I put this table to show you what are the difference between the two. Of course, the origin of the product, synthetic is coming from mostly oil and a very complex chemical system. In terms of chemical forms, as I said, it's very equal to what we found in nature. Compared to synthetic, which is sometimes different, especially for the optical isomers. It contains a very wide spectrum of carotenoids, which is not the case for, of course, for the synthetic asta, which is only containing astaxanthin. In terms of standards and accreditations, Panaferd is allowed for organic standards, and in all types of standards, especially for the major food store in the US or in Europe. For synthetic forms, synthetic asta, sometimes you have very specific restrictions. So now let's talk about some data, some results I want to show you regarding Panaferd for the two effects. The first one is related to color enhancing effect. And the second one is related to the health and health benefits of the product. So I will show you some lab results we had. The first one was made in Japan with Kuruma or Panaferd japonicus during 1 month, 30 days trials with pretty standard level of inclusions, 2.5 kg of Panaferd per ton of feed, with very controlled environment in terms of temperature, oxygen, and water quality. And you can see here, and the picture is also very significant. You see the difference between the control and Panaferd. Of course, raw, you have these blue colors, very specific of the shrimp, the crustaceanine colors. And then when you cook, you get these red colors, and you can see the main difference between the two. And again, this graph shows that the main part where the carotenoids will be stored is the shell or the exoskeleton. They're very low content of carotenoids in the muscles. Second trial, which was made in that case during a very intense and long trial we made in Thailand at the very famous Songlai University. They got a very famous aquatic lab, lab. So it was done, of course, on Vanamei in that case, with juveniles of 1.5 grams, with 6 replicates in that case, which give a more powerful and robust in terms of statistics. We got 3 diets, very standard diets. Of course, 1 control without Panaferds, and 2 levels of inclusions. First one at 0.5 kg per ton, which is a low content, and up to 1.25 in order to see the dose effect in that case. So you see here the total of carotenoids evaluated after 4 weeks, 1 month, in micrograms per grams between the head, the exoskeleton, and the muscles, and the same in terms of micrograms per shrimp. So you got, again, a very significant effect between the control, the 0.5 and the 1.25 kg per ton. In terms of colors, for the same, this is the next slide and the next results of this trial. This is interesting to see that, and it confirms the interest of Panaferd and the profile of carotenoids we find in Panaferd. There is a small content of adenirubin. So this can be considered as a sort of fingerprint. When you got adenirubin in shrimp, it probably means you have been using Panaferd or Paracoccus extract. So here you can see also the different forms of astaxanthin found. So even though it was a free form, as I said, brought by Panaferd, the shrimp has been able to directly digest and transform and esterify the astaxanthin. So you can see the cis forms, the astaxanthin form, free form, and the two esterified monoester and diester form. Yellow carotenoids are the other carotenoids found in Panaferd. Additional results of this trial, interesting to compare. Again, we made what we call the CIA evaluation, especially the redness value. Some of you are probably aware of the two value or the three value A, B, and L. So A is a redness value for the head and body, and again we can see the very significant difference between the control and the two diets. The same evaluation with, in that case, salmofan. Salmofan, you get here this table, this chart used in Asia, especially in Thailand. And you can see again the significant difference between the two, from control below, let's say, about around 20, and with 1.25 close to 22. After 4 weeks, this is a very good result. I'd like to show you a more on-field trial, in that case, coming from a farm. This is a very recent data we got from a farm based in Panama. This is a 500 hectares farm, so pretty large one, with different ponds, I would say 1.8 hectares for each. We used, in that case, 4 ponds with Panaferd and 4 ponds without, just to see and evaluate the difference. It's pretty low density, very extensive system, around 4 to 5 PL per meter square. Of course, the same type of feed. And it was quite recent. It was last month in December 2020. With very specific conditions. You know what could be the conditions during that period, especially November, December, January. You have, of course, very low temperature. And we faced what you can find, and you know what it is, the white spot disease, so very dead food problem. And this farm has also the white spot. So the trial was made in very tough conditions. And that's why it's also interesting to see in real world what could be, what type of results we may have with Panaferd. The Panaferd in that inclusion rate was 2.5 kg, so only 30 days before harvesting, before getting the shrimp out of the ponds. Initial weight was around 14 grams, and for final weight close to 20 grams. So the 3 graphs show you the results we got after 30 days with a control. This is a different type of chart, you know that well in Latin America, A2, A3, A4. So no main effect. Of course, we got very low levels of colors with a control. On the other side, with Panaferd at 2.5 kg, you can see a very high level of colors, especially you got about 1/3 of the population having A3 plus or A3 alto levels, which gives pretty good information about the efficiency of the Panaferd product. You got also about 29% of A3 medium. So you can see, and it confirms that even in very difficult conditions, which was the situation on this farm in Panama, we got good results in terms of color efficiency. So you got pictures confirming my point. So with the Panaferd 2.5 kg and with the control, you can see the very white colors of the raw shrimp, and here the very dark color, which more A3 levels in that case. Okay, we'll go on the second point, which is let's say the health interest and health benefits of Panaferd. So as you know, the main problem with shrimp that they don't have, they have only one form of immunity, which we call innate immunity. So it means this very important to get a good feed, of course, and good farming conditions. No vaccination is not efficient for shrimp. So we have been evaluating with Panaferd the effects at two levels. The first one, as some of you might know, the hemolymph effect with four main parameters widely known. Of course, the THC, hemocyte count, total hemocyte count. The second one is, of course, again, the phenol oxidase activity. We'll talk about that later on, especially in inducing melanosis effect. The third one is lysozyme, which also is a very important immune tool for the shrimp. The fourth one is related to phagocyte, and phagocytose is also a mechanism of defense, very important for the shrimp. It will, for example, affect the cell's membrane of the bacteria. The second levels of evaluation we have implemented was related to the main antioxidant enzymes. The main three are catalase, of course, glutathione peroxidase, and the third one is SOD, superoxide dismutase. These three forms will help the shrimp to protect themselves against any, especially actions of pathogens. I'll go more in details about this, this next slide, which is also another work we conducted at the Songkla University in Thailand with the same 3 diets, 6 replicates with control and 2 levels of Panaferds. And you can see clearly the statistical difference in terms of phagocytic activity between the control, the 0.5 and the 1.25, and the same for the lysozyme activities. The third slide is related to the THC counting and also the phenoxylase, as I explained to you. So again, we saw and measured a very significant difference between the control, the 0.5 and the 1.25 kg. So it confirms the immune effect, the direct immune effect of Panaferd and carotenoids contained in Panaferd, as I said, astaxanthin, adonirubin, adonixanthine, and contaxanthin. and they are inducing a very interesting immune answer. Here you have also the catalase and SOD levels measured after 4 weeks. And you can see between the control 0.5 and 1.25, the significant difference. You have here some levels of statistic expression. We have been working on a challenge. A challenge is a very good way to measure the effect of one product. We have chosen in that case 2 types of challenge. One was related to bacteria, to the Vibrio. This is a causative agent of EMS, Vibrio parahemolyticus. So after 30 days of feeding with Panaferd, we have injected Vibrio and at a certain lethal concentration. And we have measured and evaluated the mortality during 17 days in order to see what was happening. Of course, after we made a PCR evaluation to be sure that the cause of mortality was related to the Vibrio injection. And as you can see, between the control and the 2 diets, Panaferd diets, there was a significant difference. After, let's say, 8 days, you got a high level of mortality with the control, down to about 15% of survival, compared to the 2 Panaferd diets, where you got above 30% of survival, which is pretty good levels for this type of bacterial challenge. So it has almost twice the level of survival compared to the control. The second challenge was really promising, in my opinion, and made with white spot disease. After, again, the same type of protocol, 30 days of feeding with Panaferd, injection with the white spot on the 6th segment of the body with a specific concentration at the LD50. And we, of course, measure the level of mortality during 17 days. As you can see in this graph, after 5 days, all the control were dead. And you got still 10% survival at 0.5 and almost 25% at 1.25. This was quite significant. As again, I repeat, the white spot challenge is a very deadly one. So when you got such a difference and breaking of mortality, reducing the mortality level, even with the white spot injection, this is quite interesting to note. And it means that Panaferd immune benefits have helped the shrimp to sustain the white spot at least a few more days compared to the control, which is something we want to show and to see. I'd like to mention now what could be, of course, especially for farmers or for feed producers, the different strategy you may use with Panaferd. Of course, you have different situation here. I have mentioned only 4. The first one, of course, is related to color enhancing effect, which is, of course, one of the major points you're looking for. Of course, the objective is to increase the colors and, of course, increase the meat quality for the shrimp. Usually, we advise and recommend to get Panaferd at least 30 days prior to harvest. The dose is around 2.5 kg. It could be less according to the situation, and especially according to your density. The second level is also interesting to mention, especially in some countries like Ecuador or Panama or Mexico, depending on the situation, the farming situation, especially during the winter or during very high temperature, during water exchange problems. We advise to use Panaferd during the full cycle at much lower levels. It could be between 1 to 2 kg per ton. And as I show you, there is a very good enhancing immune effect. It will improve, of course, the survival of the shrimp, especially for some bacterial aspects, and you improve the final color of your product. It could be as well a sort, this is a third scenario, especially in terms of very specific conditions you may face during some cycles. It could be high temperature, it could be low salinity. Low salinity is also a major problem for some farmers. Again, we advise a 30-day, let's say 4 weeks use at a dose between 2.5 to 4 kg per ton, and it will show some interesting levels and results in terms of survival. I want to mention also a fourth situation, which is, of course, reproduction and genitors. Some of you have their own hatcheries. So, of course, carotenoids are very important, especially in order to increase the fertility, the ova quality, the sperm quality. Carotenoids are used at least 30 days prior to reproduction or mating. The doses can change from 3 to 5 kg per ton. This is something we have used in some countries, especially in Asia, where carotenoids are well known. Few words, especially for feed producers. Of course, Panaferd can be used directly in the meal prior to extrusion for some of you, or of course to pelletize, which is the main technique used for shrimp pellets. This Panaferd is quite a stable additive. So usually the loss is not higher to 10%, and usually between 5 to 7%, according to the processing conditions. Stability of Panaferd is pretty good and pretty similar to synthetic asta. Of course, Panaferd should be stored in low temperature and in avoid light. Few words about Panaferd color stability after specific conditions, especially freezing conditions, which is the case for shrimp. I'm showing here some results regarding salmon, which is, of course, one of the main users of carotenoids in the world. And this is also very important. I'm thinking about, for example, salmon from Chile, sold frozen in the US or in Europe. And we have shown that the Panaferd carotenoids are very stable in salmon, and the same for shrimp. There's no problem about that in terms of color stability. A brief final slide regarding different situation regarding Panaferd. Of course, the first point is regarding genetics. Different results may be achieved according to the genetics, according to the strains you may have. It's not the same situation in Asia or in Latin America. So of course, according to the species, final results may change. The second comment I want to show you or to give you is, of course, your target, your objective, on what market you are, Are you on more commodity and low price segment, or are you looking for more premium value? So according to that, you will adapt the level of Panaferd content. Some third comment I want to add is related, of course, to the stage. As I said, if you use at the hatchery for broodstock, or if you use at the final stage before harvesting, it's not the same target, and the amount and the content of Panaferd may change. The fourth comment is related to the feed, and especially to the feed quality. I won't talk too much about feed today, but of course, feed quality is a major point regarding carotenoids and carotenoids digestibility. Of course, cholesterol amount is important. The phospholipid content is as well important. There's very recent data showing that the fatty acid content, especially EPA and DHA, are also affecting carotenoid digestibility and transport up to the organs. So again, variations may be faced according to the feed quality. The fifth point is related to, of course, Panaferd results may change according to the time, to the timing, if it's winter time, if you have low temperature or not, of course, results may change. Of course, the sanitary situation is also tricky. It can be tricky. We have faced this during our trial in Panama. The white spot was there. Despite that, we have seen very interesting and promising results with Panaferd in that case, showing improving colors. But of course, according to your situation, you might decide to have a very low content of Panaferd or more higher in order to get both effects, the color effect and the immune interest. The final point is related, of course, to the global environment of your farm, especially, of course, the temperature, the water exchange is also, the bloom situation is also important to read and to measure and to evaluate, and may affect the carotenoid interest and benefits. There's many parameters. We could talk more in details, but I've listed the major ones here. As a global conclusions for today, I want to emphasize that the carotenoids are very important and major nutriments for the shrimp, both in terms of colors, but as well in terms of immunity and health. This is very important to note. Of course, and this is my second point, in nature, carotenoids will be found in sufficient quantity, especially if you work at low density. But now, more and more farms are using more intense systems with higher post-larvae density. So of course, feeds must provide enough carotenoid content in order to sustain both color and health. So in that case, Panaferd is a very interesting tool to get these two benefits. And again, right now, there is a tremendous trend for more deeper and red colors for shrimp. This is a case in Europe. This is a case in the US. So if you want to reach a premium segment, color is a key factor. The fourth interest of Panaferd is it's a natural form. So it's the same form that you can find in nature. Panaferd is fully authorized in organic aquaculture and it's considered as fully sustainable. It's using only fermentation process, no chemical. So it's quite interesting to underline. Panaferd's got a very specific carotenoid profile with very rare carotenoids known in the world, both in the feed, but right now more also in the food segment for their antioxidant activity and immune activities. Now, for Panaferd, we have more and more customers around the world, of course in Asia, but now in LATAM as well, confirming the interest of Panaferd for both colors and immunity. Thank you for your attention, and I'm welcoming any questions you may have.
Thank you, Dominic. It was a very interesting conference. We have some questions from the audience here. Okay. Maria Teresa Viana, it's a well-known person in nutrition, aquaculture nutrition in Mexico and Latin America. She is asking about in extensive farms, how to know how much they will really need the carotenoids, I mean the level of if in extensive farms if it should be used the total recommendation or less than the total recommendation using it.
Yeah. Okay. Thank you, Salvador. It depends again on the target on the objectives of the farms. And of course, you need to make a sort of audit of the situation. What are the actual results of the farm in terms of color, in terms of sanitary situation? If the level is pretty low, there is something to do. And of course, Panaferd can be added, at least at the low levels. If, as I mentioned in the different strategy, it could be 1 month prior harvesting, or it could be during the full cycle. So there's no definitive answer to that question. It depends on the target of the farms. I would say, as we show here, for example, in Panama for this trial, but I could show you more, we use only 2.5 kg per ton during 1 month, 4 weeks. And it gives, compared to the control, and you can see it was extensive system, 5 PL per meter square, we got significant results. And the farmer was very pleased. So that's also the final conclusion. The farmer was pleased by the results, and it was sufficient for him.
Okay. Another question from Ed Aguirre. He wants to know if he compare with our source of astaxanthin or any other carotenoid like that or similar. How is the cost for use this product and other source of astaxanthin?
Well, I'm not involved in that part of the cost of the product, but again, just a few words about it. When you use this type of product, you place your product on the premium segment. So the additional, of course, price you may get from that is also interesting. So it's gonna represent few cents per kg in any case, and the extra price will cover the cost of your Panaferd product. Maybe Carolina, you want to add some points about it?
Sí, hola. No sé si estoy al aire o no.
Sí, sí.
Escuchamos. You can speak in English if you want, because this is a English recording and the translator.
Prefiero hablar no tengo que hablar en español porque si no, no vamos a que me pongan directo, no sé.
Sí, sí.
A ver, con respecto al costo para los camaroneros en Latinoamérica, hoy no tenemos un precio definitivo porque el producto se va a comercializar a través de las plantas de alimentos, entonces cada una puede de repente cambiar un tema del precio que ofrece. Pero si se da la misma situación, hoy el producto ya lleva 12 años en el mercado de salmones. Entonces, si lográramos, o sea, en el fondo, si el precio va a ser muy similar a lo que se maneja en salmones, basado en las experiencias que hemos trabajado con camarones, podríamos decirte, no sé, capaz que pueda salirte en una buena situación normal de equilibrio, un promedio, 2-3 centavos de dólar por libra de camarón, que por ejemplo nuestra experiencia en Panamá a nivel de costos nos salió, tuvimos mala conversión por un tema de que tuvimos baja efectividad de alimentación, perdimos un poco de alimento, pero tuvimos, no sé, 4 centavos por libra de costo, pero el extra que se sacó por tener una 3 en vez de una 2 es de 15 centavos por libra de camarón. Y lo otro es que uno sacando las cuentas, básicamente en cuanto al potencial de mejorar la sobrevivencia del producto, con un 4% de diferencia de mejorar la sobrevivencia ya se paga. La verdad es que el Panaferd en sí lo que tiene, como es tan concentrado en carotenoides con muy poca cantidad, si tú eres eficiente en la alimentación, no se nota. ¿Qué será? No sé, menos de un 1% de tu costo. El costo y principal va a venir de cuánto eres eficiente con tu alimentación y cuánto controla su producción. Panaferd puede ser muy barato en sistemas súper intensivos porque sabes que todo el producto se lo consume. En sistemas extensivos, en donde aparte tienes la competencia del alimento natural, es mucho más difícil de predecir un costo, porque no hay control sobre la producción. Pero en el fondo, la situación del costo es mucho menor que en salmones y no tiene no es muy, se compensa fácilmente, y lo bueno que tienen en camarones es que hay mercados que pagan más por más color, que no lo tienen en salmones. No sé si eso es lo que le podemos decir por ahora. Más adelante ya vamos a poder tener más claridad de estrategia y valor exacto, ¿no?
Indeed, this product could be used coated. So with some oil usually, or any type of lipid form, usually fish oil or any vegetable oil, but this is possible. Again, if Panaferd is coated, you need to be sure that shrimp will eat in a very short period of time, because of course the feed will release part of the oil and you might lose the carotenoids in the water. So if your feed quality is good enough, if you have a good coating activity process, you may use Panaferd as a coating agent. Yeah, it's possible. Salvador, I want to insist on one major point, and could be my conclusion here. Panaferd has two benefits here. It's not like synthetic asta. Of course, synthetic asta is known from the 90s. We are talking about a natural color, natural product, having two benefits, the color enhancing effect and the health effect. This is quite new, actually. We are discovering the past, let's say 5 years, a lot of research around the world have been conducted to evaluate especially the antioxidant effect of carotenoids, especially astaxanthin, adonirubin, and adonixanthin. And in the food sector, especially for human health, there's major works in order to use the properties and the benefits of this type of carotenoids. So the approach and the strategy with Panaferd is to get the same. Okay, you need to increase the color. There is a demand for that, especially on the premium segment. But we know the situation of, and especially the sanitary situation of farms in Latin America. Sometimes it's very bad. It's very bad. The survival is very low. So the only way you can improve the farming results is by using this type of new additives and having a nutritional strategy, colors and immunity in that case. So Panaferd has the benefits to get a very wide spectrum of carotenoids. It's not only astaxanthin. That's the main interest of the product. So it depends on the density you're using. Are you working at 5 pl per meter square, or are you working at 100 pl per meter square? This is the main difference. So of course, if you use 100 pl per meter square, you need to enhance color and promote the immunity by different nutritional additives, what we call functional feeds now. So you may have functional feeds with different approach, including probiotics, including, of course, fatty acid content. You have, of course, the nutritional approach. Panaferd is within this functional feed. So this is a type of strategy now growing in Asia as well, especially in Thailand.
¿Escuchan a mí? Ya, sí, perfecto. Sí, estaba que estoy con los dos audios, sí, disculpe. A ver, las bolsas Panaferd viene, bueno, en el formato de polvo seco, en cajas de 20 kilos. Dentro de la caja de 20 kilos, adentro vienen 2 bolsas de 10 kilos en un papel, bueno, en un material aluminizado, al vacío, básicamente para conservar todas las propiedades del producto. El producto se fabrica por fermentación con 0 químico. El único aditivo que lleva es carbonato de calcio para la mejor uso. Y no se aplica ningún antioxidante, nada, y eso va, por eso vienen bolsas de vacío, aluminizadas, protegidas con la caja. Básicamente, y también hay, bueno, en Europa se ocupa principalmente los salmones, ya vienen en formatos de 500 kilos en maxi, en donde los colocan en los hilos y ahí se aplica. Pero básicamente ya en la producción más industrializada en salmones, ¿no? El producto, no sé si lo dijo Dominic, pero ya Panaferd ya tiene más de 12 años en el mercado. Es muy fácil usarlo, la verdad es que conocemos bien la cantidad de carotenoides, es muy fácil dosificarlo. El gran desafío con los camarones en Latinoamérica es básicamente que hay un abanico muy grande de diferentes tipos de producción, y cuando tú no controlas, esto, bueno, yo llevo bastantes años en la acuicultura, en salmonicultura, y claro, todavía a la parte de producción de camarones le falta un poco más de control. Los que están intensificados, que ya saben cuántos camarones hay, mantienen la calidad de agua y tú le das de comer y se comen todo el pellet, tú puedes dosificar mejor el Panaferd, con muy poco puedes lograr resultados. El desafío es en la parte extensiva, en donde no hay muy buena alimentación automática, en donde no se controla bien todas las variables, es difícil dosificar. Y lo otro que hemos notado en la prueba en Panamá, cualquier cambio de estrés, el camarón lo primero que hace es perder el color. Entonces tú puedes tener una semana antes de la cosecha un camarón a 3, listo, está perfecto, tuviste un cambio de pH, alcalinidad, lo que sea, y el camarón lo primero que perdió es nivel de color. Y lo que nos sirvió Panaferd en Panamá, que tuvimos justamente ese problema, es que nos ayudó a mantener el color durante los días antes de llegar a cosecha y no perderlo. Los controles perdieron color, nos bajaron el color, y ahí la empresa perdió plata, en el sentido de que ellos tienen, con más color, logran mejor precio. Y el otro punto es el tema de la sobrevivencia, que acá, en cualquier producción, la única manera de compensar los costos es tener buena calidad del producto y buena sobrevivencia. Y para tener buena sobrevivencia usando Panaferd, tienes que también asegurarte que se lo coman. Hoy hay un montón de aditivos positivos para la salud, pero si no tienes la eficiencia en sistemas extensivos, donde tenés el alimento natural, que es el preferido por el camarón, y tenés que darle un pellet, y querés que el camarón coma el pellet en poco tiempo, y tratando de que el alimento sea barato, y que todos esos aditivos que lo coloquen les den un beneficio, tienes que hacer que se lo coma. Y yo creo que el gran desafío para Latinoamérica es lograr una buena eficiencia y una buena calidad del alimento para que cualquier ingrediente aditivo positivo puedan captar los beneficios y mejorar la calidad del camarón. Lo bueno es que Panaferd va de la mano de todas las tendencias, no se usan recursos naturales para producirlo, es sustentable, aceptado por el mercado orgánico. Entonces, todas las empresas que van hacia un valor diferenciado, a tener sustentabilidad, hacia donde van todos los mercados, con Panaferd lo pueden lograr. Al tener carotenoides que son muy potentes, con muy poco logras mucho. En cambio, si usas otros carotenoides de menor calidad o de menos nivel antioxidante, tienes que estar hablando de poner porcentajes en tu dieta muy grandes y más difícil de compensar. Y el camarón tiene que metabolizar un carotenoide que es, no sé, producto de origen vegetal y transformarlo en el que necesita. Acá con Panaferd es lo mismo que se come el zooplancton. Paracoccus carotinisfacies está en el fondo del mar, lo comen los zooplanctons y los mismos camarones cuando comen barro, filtran barro, también lo consumen. Es exactamente el mismo componente que obtienen de la naturaleza y va directo, no tiene que hacer ningún trabajo de metabolizarlo ni nada, y con poco tienes alto nivel antioxidante. Esa es una gran diferencia cuando tú ocupas antioxidantes de plantas, en donde, claro, tienen su color y tienen su nivel antioxidante, pero están acá abajo comparado con el otro acá arriba. Nosotros tuvimos 4 lagunas con Panaferd y 4 lagunas control. Tuvimos problemas ambientales y en las 4 lagunas con Panaferd mantuvimos o mejoramos el color, y en las 4 lagunas controles perdimos color, porque ante un momento de estrés, lo primero que hizo el camarón de las que no tenían Panaferd es ocupar el color que ya tenía. La astaxantina es el primer recurso para defenderse. Fíjate que todos los huevitos, todas las ovas tienen ese color naranja porque la naturaleza eligió los carotenoides para defenderse de los cambios ambientales, los rayos UV y todo eso. Entonces, lo que hace el camarón, lo primero que hace es usar sus carotenoides. Con Panaferd tú haces, que te das un nivel de aseguramiento de que cuando estás llegando a la cosecha, mantener tu biomasa. Le das Panaferd, asegurás que se sobreviva la mayor porcentaje de tu biomasa que tienes ahí y que mantenga el máximo de color. Es como un seguro de tu biomasa. Uno ve los costos y a partir de los 7 gramos para arriba el consumo de alimento, todo eso es lo que te afecta al final el costo final. Que tú no pierdas biomasa grande es re importante, y ahí Panaferd te puede ayuda, que es lo que estamos viendo. El mayor desafío es lograr estandarizarlo en sistemas que no tienen mucho control. Eso yo en intensivo, súper intensivo, le digo a ciega, ponele tantas semanas todo y vas a encontrar mejora seguro, porque tú controlas el sistema. Pero en extensivo hay que buscar un equilibrio, quizás una dosis alta con pocos pellets, pero asegurándote que esos pellets se lo coman, como decirte el suplemento diario. Tienes que asegurarte que el camarón lo coma, porque al final el camarón va a tener la ayuda si tiene una concentración de carotenoides para ese gramo de peso, si lo ingiere. Es como que tú te dieran vitamina C, pero no te tomas todo el jugo, sino un traguito. No vas a tener ese efecto. Básicamente es eso, hay que entender que estamos tratando con organismos vivos. Y nada es mágico, y hay un montón de variables. Ese es el tema de la producción de organismos vivos.
Para aclarar un poco más la idea, extensivo, ¿a qué te refieres con extensivo? ¿Qué densidad de siembra más o menos?
Por ejemplo, extensivos fue el caso que hicimos en Panamá. Teníamos menos de 6 camarones por metro cuadrado, en algunas lagunas 3 camarones por metro cuadrado,
es muy extensivo eso.
Y 2 alimentaciones al día, un viejo remando y tirando sin... Bueno, yo vengo de la salmonicultura donde hay un cálculo de pellet por camarón, acá no, o sea, es que es todo, y que esas 2 veces de alimentación no compensa mucho porque en 35 minutos el alimento se nos desarma y ya después el camarón no se lo come. Entonces al final perdimos mucho producto por tratar de que comieran el máximo, pero básicamente por una calidad del alimento, de la lixiviación y demás, se pierde. Entonces hay que mejorar la cantidad de veces, hay muchas cosas que aprendimos, pero a eso me refiero con extensivo, que de repente no es eficiente y que hay muy poco camarón y que el camarón tiene mucho alimento natural que es el que prefiere. Si el camarón se alimenta de zooplancton y de la laguna, no va a lograr la misma concentración de carotenoides que si comiera un poquito de Panaferd, porque la cantidad de carotenoides en un zooplancton es menor de la que va a tener el pellet.
Ya muchas granjas en Ecuador y en México son más intensivas y muchas están
Claro.
utilizando alimentadores automáticos para estar alimentando más frecuentemente y que el camarón se coma el pellet. Entonces yo creo que ahí va a haber muy buenos resultados en ese tipo.
Hacia eso vamos, y yo creo que la industria del camarón va a acercarse un poco a lo que terminó haciendo la salmonicultura. En el fondo tú tienes que tener control de los costos más grandes están en el costo fijo, el personal y el alimento. Tú tienes que tratar de buscar un equilibrio y de controlar esas variables para lograr tener realmente buenos costos. No está en bajar la calidad del alimento, no está en bajar las cosas positivas que uno le da, porque acá el camarón, si come y come todos esos beneficios, logra tener inmunidad y logra sobrevivir. Si no come bien y no sobrevive, o un camarón enfermo que le quieras dar Panaferd cuando ya está enfermo para que se recupere, no, porque no va a comer.
Muy bien, pues no tenemos más preguntas, bueno, hay más preguntas, pero tenemos ya el tiempo limitado. Dominique, thank you very much for your presentation. Very interesting topic. Carolina, muchas gracias. Por participar en este evento. Las preguntas que vengan llegando se las vamos a hacer llegar poco a poco. El webinar va a quedar en la página del canal de YouTube de Panorama Acuícola y ahí vamos a estar captando la información y preguntas las vamos a hacer llegar. Muchas gracias. Dejo el micrófono en manos de Claudia, que va a dar algunos mensajes y a despedir la transmisión. Gracias.
Muchas gracias. Merci.